Hello. We're going to continue this hemostasis video. We're going to go into part 3. Now, we basically finished up talking on the basis of the vascular spasm, and we talked a little bit about the platelet plug formation. We're going to continue. Just to kind of recap, make sure you guys remember everything. Hemostasis, so again, what is hemostasis? Hemostasis, hemo means blood, stasis means stop, so it's a blood stoppage. It's a fast localized event that occurs in 3 phases. What are those 3 phases? The first phase is going to be vascular spasm. It's the first immediate event that occurs within an injury or damaging or rupturing a blood vessel wall. Second thing that occurs is platelet plug formation. And we talked a little bit about some of the chemical methods that occur in platelet plug formation, but vascular spasm again. We talked about vascular spasm. What is vascular spasm? We said that there was 3 events that take place in vascular spasm. Okay, the first event that takes place in vascular spasm is chemicals. And I'm just going to put chemicals. We talked about that. Just to recap, just to make sure you remember, chemicals. Remember, endothelin was a chemical that actually is released by the injured endothelial cells, and basically what it does is it helps to be able to bind onto the smooth muscle cells and enhance the calcium entry into the smooth muscle cells. Because you know, as you enhance calcium into the calcium entry into the actual muscle, it's going, especially in smooth muscle, it's going to activate a intracellular regulatory protein called calmodulin, and then that activates a myosin kinase. Myosin kinase phosphorylates the myosin head, and that triggers the contraction mechanism. But there's that, and then there's thromboxane A2, which is a derivative of prostaglandins through the, we have what's called exonoids. Exonoids are biologically active lipids. You'll learn this in the endocrine system, but they're biologically active lipids formed from another molecule called arachidonic acid. They don't really act as true hormones because they're highly localizing in their effect. They're not widespread. But there were thromboxane A2, and there was also another chemical called serotonin, which helps to be able to enhance it. We also said that it could be direct injury to the vascular smooth muscle, and we call it a myogenic mechanism. And the third thing we said, so let's put that in there, we said it could also be a myogenic mechanism because of direct injury to the vascular smooth muscle. And the third thing was basically activation of pain receptors. We talked a little bit about that. We're not going to, maybe in future videos I'll go through ascending and descending pathways, but it's just activating the pain receptors within this area and then sending those impulses up to the medulla. And then the medulla has a center called the vasomotor center within the medulla. If we just briefly look at that, I'm not going to, you don't really need to know all this crap, but if I take the central nervous system, now you have the cerebral hemispheres, come on, midbrain, pons, medulla, spinal cord, cerebellum. Right here is going to be the midbrain, right here is the pons, and then you have the medulla. There's a nucleus here, and in future videos on blood pressure regulation we'll talk about it. That nucleus is called the nucleus of tractus salutaris, and then there's another center, and that center is called the vasomotor center. And the vasomotor center basically just helps to be able to send impulses to the vessels to be able to enhance vasoconstriction. But we'll talk about that in future videos, maybe on blood pressure regulation. Okay, but that was basically it. Now we're going to talk about platelet plug formation. So let's pick up where we left off. Now, we talked briefly about this. We know that healthy endothelial cells, intact endothelial cells, smooth endothelial cells release chemicals that help to inactivate the platelets. And those are chemicals like prostacyclin, PGI2, nitric oxide, and then adenosine diphosphatases. Well, let's figure out what it releases. Obviously, it doesn't release those chemicals now that it's not healthy. It releases another chemical. We talked about one of them, endothelin, but there's another one. What I'm going to do here is you guys should know by now that if you study the tunics of the blood vessels, arteries and veins, that there's an endothelial layer, and then the endothelial layer, which is the tunica interna, has a subendothelial layer, which is made up of loose connective tissue, and that's collagen fibers, basically. Well, what happens is these endothelial cells release a very important chemical. This is a very, very large plasma protein. What is the name of this plasma protein? The name of this plasma protein that these injured endothelial cells release is called von Willebrand's factor. So the injured endothelial cells make a plasma protein, they synthesize it and release it, and what this protein does is it binds onto the collagen fibers. So it binds onto the collagen fibers, it sticks to the collagen fibers. And now it has little points on it, little receptor points, little amino acid ionized polar regions on the other side. And so basically it forms this cross bridge formation between the collagen fibers, and guess what? You guys remember I told you that the platelets were kept inactivated because of the prostacyclin, the PGI2. You know, they were sleeping, they were crying too, you know, because they couldn't become activated because they were continually kept inactivated by PGI2 nitric oxide adenosine diphosphatase. What happens is they don't release those chemicals, and because they don't release those chemicals, he becomes happy. That frown turns upside down, and he becomes educated. He grows some hair, gets a pair of glasses. You know, he becomes smart. He has ears, you know, a big nose too. He becomes educated, and he even has a little bow tie, okay, because he becomes smart now. So what happens? This platelet starts to adhere to the von Willebrand's factor. Because what it sees is it sees the collagen fibers. It doesn't see anything, but it just actually wants to adhere to the collagen fibers. But when it comes to adhere to the collagen fibers, there is a protein called von Willebrand's factor, which is released by the injured endothelial cells. So basically what begins to happen. So now, this platelet comes, I'm going to draw him green now. This platelet comes in, binds onto that actual von Willebrand's factor. And when it binds onto the von Willebrand's factor, this protein part, this amino acid polar ionized region, activates the receptor on this platelet. And when this receptor on this platelet is activated, he has granules in here. You know, there's different kinds of granules. I'm not going to go into the different kinds of granules because there's a whole bunch of different kinds of granules and specific names for them. I'm not going to do that. Basically, what we're going to do is we're going to say that there is granules, and these granules, whenever this von Willebrand factor activates this platelet, the granules start to become stimulated. They undergo degranulation. And when they undergo degranulation, they release three chemicals. One is adenosine diphosphate. What the frick is adenosine diphosphate? What's important about it? We'll talk about that. What's the next one? He releases another chemical called serotonin, which is a biogenic amine. And he releases another one called, let's draw these bigger so you can see them. One of these is called adenosine diphosphate. I'm not going to write the whole name, you guys should know how to spell it. Adenosine diphosphate. The other one is serotonin. And the other one is called thromboxane A2. Okay, let's explain these chemicals. Okay, so far we've known injured endothelial cells release von Willebrand's factor. Von Willebrand's factor binds onto collagen fibers, forms a cross-bridge formation between the collagen fibers and now the aggregating platelets. The platelets begin to adhere to the von Willebrand collagen complex. The receptors on the platelets are activated by the proteins. The proteins then, when they activate the receptor, there's receptor sync chemical intracellular signals that activate granules. The granules begin to degranulate and release three potent chemical messengers: adenosine diphosphate, serotonin, and thromboxane A2. Now we just got to figure out what the frick do these chemicals do. Well, adenosine diphosphate is a potent aggregating agent. What does that mean? All these big words. It's a potent aggregating agent. Basically, to make it simple as possible, it acts as a chemical or I could say, let me say it like this, a chemotactic agent. Let's say it like that, it sounds pretty good. What it does is it attracts in other platelets that are circulating through this plasma of where the blood vessel injury is occurring. So those chemicals, ADP, serotonin, thromboxane A2, start causing platelets to adhere to the site of injury, where the blood vessel was ruptured or damaged or underwent severe trauma. Okay? When it does that, ADP just causes them to adhere, but not only does ADP cause the platelets to adhere, but it'll then cause the platelets to be able to release their contents. Because why? Because now another platelet will start rushing here. His receptors will get activated. Another platelet, another platelet, another platelet, another platelet, another